Okay, hello. We're going to go ahead and get started. And so if you're still grabbing food or anything like that, that's fine. Take your time getting seated. But welcome to the Modern Research Technique Series. So this is a collaboration between Roberts and the University of Rochester Medical Center. And it's sponsored by a grant from the National Institute of Health, which is a training grant called T32 Training Grant. I'm Morgan Monahan. I'm a postdoctoral research associate at the University of Rochester Medical Center. And I also graduated from Roberts from the biochemistry department in 2011. So I'm really excited to be back and be bringing students from the U of R here in this partnership with Roberts. So I want to thank you for hosting and thank you for coming. And what this partnership allows us to do is that we take students, graduate students, PhD candidates, and postdocs from the University of Rochester Medical Center here to practice their teaching in front of undergrads. And then in return, you hopefully learn some things about some modern research techniques, and we will feed you lunch too. So that our presenters can get feedback on their presentations, we ask that you grab, if you haven't grabbed them already, 2 evaluation forms from the front and just evaluate the presenters on how you feel that their lesson went. And we encourage faculty and staff here, if you would also fill those out too, we would really appreciate it. And then when you're done, if you don't mind, just there's a box, an empty box up where you grab them, just stick them back in the box, and we'll make sure that Lisa and Dahlia get their evaluation sheets. Also, if you haven't signed in yet, if you don't mind signing in, we need to keep a record for grant purposes of how many people that we've fed. So each presentation is going to be 20 to 25 minutes. If you have questions, feel free to interrupt, raise your hand, stop the presentation, and kind of raise your hand and say, I don't understand that. And there also will be time afterward for questions, so if you have a question, just ask it whenever you want. So I think that covers pretty much everything. I'm going to introduce Lisa, who is our first presenter. So this is Lisa Houston. She's a PhD candidate in biochemistry and molecular biology at the University of Rochester Medical Center. And today she's going to be talking about flow cytometry and fluorescence-activated cell sorting.
Thank you, Morgan. So I wanted to kind of make you even more excited to be here, and I brought some M&Ms. But before I pass those out, I kind of want to talk to you about M&Ms. And so I have here a mixed bag of M&Ms. And there's caramel M&Ms and peanut butter M&Ms and peanut M&Ms and plain M&Ms, and you guys can pick which ones you want. But it's really kind of hard. I really like the peanut butter M&Ms. They're my favorite. But I'm not really sure how to tell which of these M&Ms are peanut butter M&Ms. So does anyone have any ideas of how we can determine which M&Ms are the ones that we want?
Crack it open and look inside.
Okay, crack it open, look inside, or take a bite. What else? How can I mean, if you take a look at these, it's pretty obvious, I think, which ones are the plain M&Ms. They're all about a certain size. And the ones that aren't plain are a bit bigger. So we could possibly look at the size and be able to determine. I don't know how accurate I would be. I think this one might be a peanut M&M. Are there any other ideas? No? All right. So of course we can use the size. Perhaps you want a particular color. You don't like the red M&Ms or something, so you can use the color to sort them. And we can also use the components, which in this case, we'd pretty much have to bite in to see. So I'm going to go ahead and let you guys pick your M&Ms while I continue with my lecture. So just kind of make sure everyone gets a chance to have those. All right. So how can we, similar to finding out my M&Ms from my mixed bag, how can we determine which cells are the one we want? So say we have a mixed population of cells, like in a blood sample. We can use the same idea. We can look at the components, the size, or even the fluorescence or color of the cells in order to determine what they are. And this is the idea behind flow cytometry. And flow cytometry occurs in a machine like this. And even though this looks like it's pretty small, it does fit on a table, but it's more like would take up about 2/3 of this lectern stand. And it's probably about 2.5 to 3 feet in height. So it's a pretty big machine. And you take a tube that is about this big. So similar to what the blood is collected in. And this is your sample. And you insert this into the machine right here. And it will take the sample and it puts it through the machine. And this is kind of what the inside of the machine looks like. There's lasers, there's mirrors, there's detectors, there's tubes. You can't actually really look in the machine because of the lasers. So flow cytometry uses laser light to characterize cells. And this is kind of a schematic of what the idea behind flow cytometry is. So you bring your sample in, and using hydrodynamic focusing, cells pass through in a single file. So you can get very small sample, and you should be looking at one cell at a time. Depending on your flow rate, though, you may get doublets where you have cells that are kind of combined together. And then you have the laser light source that shines and hits the cell, and from this you can get fluorescence emitted from the stained cell, or you also get forward and side scatter light that comes directly from the laser light. And the type of light that's reflected or emitted from the cells is detected and determines the size, composition, and fluorescence of the cell. So we're going to go a little bit deeper here. Forward scatter light is what's traditionally used to determine the size of the cell. And forward scatter light is refracted by a cell, and as it continues, and the light itself continues along the light path, which is the same direction that the light was originally traveling. So that's kind of demonstrated here. Here we have the light path, it hits the cell, and we get this forward scatter. And so the larger the cell is, the more forward scatter. And the machine is set up so that if you have a small cell that doesn't take up the entire light path, it doesn't detect the original light. It will only detect the scattered. So it will only get the forward scatter. So the smaller the cell is, the less scatter light there is. And the next component that it looks at is side scatter, and this is used to determine the granularity or the complexity of a cell. And side scatter light is refracted by the cells in a direction that is outside the original path of light. So it angles it off to the side. And the more components and things that you have inside the cell, then the greater this side scatter is. And so if you have something like neutrophils that have high complexity, you will produce more side scatter light. So what you get after you run a sample through flow is you get this scatter plot. And each one of these dots represents a single cell. So in a matter of 30 seconds to maybe 10 minutes, depending on the saturation of your sample, you can look at 10,000 cells. And you can determine what those cells are. So you get this scatter plot, and on the y-axis is the side scatter, and on the x-axis is the forward scatter. And so here they've labeled some of these cells for you. And so D are considered degrees. They have debris. They have very small forward scatter and side scatter. So they're really small cells or particles. They may be some salts or things that are in your solution. They're just debris, parts of cells, not really what you're looking for. Here, what they've labeled L or M are considered leukocytes or monocytes if you're looking at a blood sample. And these are small to medium cells with low internal complexity. So they have medium forward scatter. You can see they've moved up on the x-axis from where the debris are, and they have low side scatter. They're not that much more inducing of side scatter. And finally, you have what they've labeled G here as granulocytes. These are large cells with high internal complexity, high forward and high side scatters. All right. So to even make sure that you really know what you're looking at, another component you can add is you can add fluorescence. And this can help increase the accuracy of cell identification as long as adding other things for you to look at. So a fluorescence molecule is excited by a compatible wavelength laser, and the light emitted by the fluorescence molecule is detected, which is kind of shown here. So again, we have the sample comes in, and due to that hydrodynamic focusing, we get a single cell that comes through, and this laser light shines on it. And here you can see that we have the forward scatter detector, so we again are still getting that forward scatter and side scatter. But then we also have a way for it to get the fluorescence that is emitted and it uses this beam splitter to separate lights according to the wavelength. So from a single cell, you can get more than one type of fluorescence. Typically, if I don't look at blood, but the samples that I take in, I have tagged with either with both a red fluorescent protein and a green fluorescent protein. And from one single cell, I can get the read of how much red fluorescence I'm getting and green fluorescence at a single time. And fluorescent molecules can be naturally occurring. There are certain amino acids that fluoresce. They can be genetically added to a protein. That's usually what I have, is I've tagged something with the GFP genetically. Or you can have antibodies that fluoresce that are bind to antigens on cell surfaces. So this is an example of a histogram that is also some of the information you might get back as you run a sample through the machine. And on the y-axis, you have the cell count, and on the x-axis, you have the fluorescence. So these come from a population of, I believe they're T cells, and they have been tagged using the antibodies. And some of the, they're looking here for CD8. And some of these cells don't express CD8, and some of the cells do express CD8. And the area under each peak is proportional to the number of cells in each population. So you can see that in this sample, they had more cells that were expressing CD8 because the area under the peak is much larger than those that were not expressing CD8. And as you move along in the right to the right on this, I don't know my rights from left very well. So as you go along the right, you get a higher amount of fluorescence. All right. So to kind of summarize a little bit about what flow cytometry is commonly used for, it's used to identify antigen expressing patterns on cells. It's also used in gene expression and transfection efficiency. So it's a way for people who use techniques like RNAi or CRISPR to silence, to ensure that their gene has been silenced. It's really commonly used for immunophenotyping. In fact, most of the, in the floor core at the University of Rochester, most of what is done is analyzing blood. So they can analyze bone marrow, cell lines, platelets, and this is clinically used to diagnose hematological malignancies such as lymphomas and leukemias, which is a much faster way to do it than what is traditionally done. Or when you used to go to the doctor when I was a kid and they would take your blood and then somebody would actually have to count the cells in your blood. It also can be used for cell cycle analysis, so using fluorescent dyes to measure the 4 distinct phases of a cell cycle. And you can measure aneuploidy associated with chromosomal abnormalities. So again, instead of having to go through and do staining and physically look at a whole bunch of hundreds of cells to try to determine if a person has an aneuploidy, you can quickly screen them and in a matter of seconds know the results. It can also be used to determine apoptosis. So apoptosis is programmed cell death, and it usually doesn't involve an immune response, and so you can use flow cytometry to distinguish between apoptosis and necrosis. It's done with cell proliferation assays, which measures cellular metabolic activity in response to stimuli, and it's also used for what's called intercellular calcium flux, which is activation or signal transduction pathways and the cell's response to them. So to really get a good understanding of what cells you're looking at, one of the things you can do with flow cytometry is called gating. And gating selects populations for further analysis. So gating is deciding, and it's done by the user. They decide which cells meet certain criteria that they want for further analysis. And gates are drawn so that the targeted cells fall within the gates. And then these gates can be based on forward scatter and side scatter or based on fluorescence. So I have an example of an experiment which I ran. And on the y-axis, we have the side scatter, and the x-axis is the forward scatter. And you can see I've placed a gate around the cells that are of interest to me. They have the size and complexity that I want. This is also done with blood. And so this is using the forward scatter and side scatter, and they used to identify a cell type. And here, these were identified as lymphocytes. So when you have it on your computer screen, you have this blank gate with nothing in it. When you run your samples, the cells populate on the forward scatter and side scatter, and you've already identified where you've already set the gate, and you said that most of the cells that fall within this area are the cells that I want targeted. And you usually set your gate so that you get somewhere about 90% of all of the lymphocytes should fall within that gate. And so that's what happens here. And then you can further using, in this case, they used fluorescence that was antigens tagged to certain antibody tagged to certain antigens that they wanted to look at. And they were able to, from these lymphocytes, determine which ones are B cells and which ones are T cells. And this becomes important when you look at fluorescence-activated cell sorting. So fluorescence-activated cell sorting, you physically sort populations of live cells. And so you can use the forward scatter and side scatter and the fluorescence data collected at flow cytometry. The user defines the characteristics determined on how the cells should be sorted, again, by setting those gates. Sometimes they might be set up more as like bins, and so they might be referred to as bins. And then the fax machine uses electrodes to charge specific cells. So it can give them a positive charge or a negative charge, or more complex ones can give them partial charges, so that you can sort an abundance of different parameters. And these electromagnetics then sort the cells, and they're charged into separate collection tubes. And so you're now collecting live cells that have been in their buffers or within the solutions that they need to keep them live. So that allows you to then take these cells and further analyze them, which is the key with fluorescence-activated cell sorting. So FACS is routinely used in research and pharmacological and biotechnology companies to isolate cells for further analysis. And some of the things that this is used for is they might do, it's like high throughput screening. So they can do screening of peptide libraries and look for those that bind, and then they can then collect those cells and discover what characteristics of those cells allowed them to have this binding. It's used for selecting antibody mutants, which is influential in vaccine development. It's screening for enzymatic activity and also for isolating specific cells just for further research. So like once you've collected these cells in a sample like this, you can then take the cells and that are the ones that are of interest to you, and you can do like high throughput genomic data and determine what genes are upregulated or which proteins are really expressed well in it. And so my last couple of concluding slides here are just some recent developments with, more recent developments with flow cytometry. And one of the new things that machines and new things that are going is called imaging flow cytometry. And here we characterize and image hundreds of cells in a second. And so this is really good for determining things like the co-localization of two proteins or morphological changes that may happen. So you can fluorescently tag two proteins, and if you want to see if they are co-localizing, you can run through, and like I said, it gives you hundreds of images of individual cells. And here you can see that you can look at specifically, like I want to see where the green fluorescent protein is expressing. Here I can see where the yellow fluorescent protein is. And then I can co-localize it and see that this red fluorescent protein and this green fluorescent protein really seem to be together. And so that can give you an idea that these proteins may be interacting. And the last thing that I've found about flow cytometry is what's called mass cytometry. And I really don't know a whole lot about it, but it's labeling. What you do is you label the samples with antibodies that are bound to metal isotopes. And so it's kind of combining mass spec with flow cytometry in that you have these antibodies that you go through and allow for separation. And this allows you can collect information on 27 different parameters for a single cell. So that can give you some really in-depth information about what is going on with your population of cells. The one drawback to this is that if you use this technique, you can't collect the cells afterwards because they're destroyed in the experiment itself. So that's kind of what I have for you. I hope that this Gives you a good idea of flow cytometry. Does anyone have any questions? Perfect. Okay. I do. Okay, so my research is in the yeast Cerevisiae, and so what I have been studying are what's known as inhibitory codon pairs, or pairs that of codons that, when they're present, actually reduce the expression of a protein. And so what I have is what's called the RNAID reporter, where I have a bidirectional promoter, and upstream of that bidirectional promoter I have the red fluorescent protein, and then downstream of that I have inserted my inhibitory codon pairs, and I have the green fluorescent protein. And so I can measure the expression of that green fluorescent protein based upon whether the inhibitory codon pairs are there or the synonymous optimal codon pairs are there, and I'm able to normalize that for each cell based upon the red fluorescent protein. So if, for example, if a cell was just overexpressing and I got high levels of GFP, if I would expect to see a high level of RFP also. So that allows me to normalize and to ensure that the only difference is what's going on with my inhibitory codon pairs. So that's what. Thanks. Anyone other questions? Okay, thank you.